Okay, good afternoon, everyone. And we're going to carry on our discussion about viruses. And I'm going to actually revise part of our last lectures. I know that some of you don't have all the slides I'm going to use, so I will put these up again for you. And I know that there is some confusion, particularly about RNA viruses and the different kinds of genome that they have and what that implies for when they want to replicate themselves. So I'm going to go over that, and I've added a slide here. So first of all, remember what I told you. The viruses need to do two things. Once they're inside the host cell, they need to get the host cell machinery to replicate their genome, the viral genome. That's number one. They need lots of copies of the viral genome so that every new viral particle that is constructed has its own copy of the genome. The second thing that they need to do is to make all the viral proteins. So what they're going to need to do is take over that whole translation machinery of the cell, and they're going to have to get, instead of the host producing its own proteins, the host cell is going to start producing viral proteins. The viral proteins package themselves together into the capsid, and the capsid is packed with the genome. In this case, we're talking about the RNA genome. I will mention DNA genome just to put it in contrast. But the RNA genomes are quite variable depending on the virus that you're talking about. So we have heard of three kinds. The first are the positive strand RNA genomes. And the second were negative strand genomes. And the third are positive strand genomes, which actually need to be turned into DNA in order to function. So those are the three kinds. The last one are the retroviruses. But I want to just briefly discuss the positive strand and negative strand RNA viruses, just so that it's absolutely clear what the difference in their functioning is. So here's how it works. We're going to first of all just think about a positive strand genome. So this is a single strand RNA, and here it is. I've just given you a little short bit here. From this, we can actually replicate this. We can transcribe it so that here we have, just by matching base pairs, here, look, A, U, G, C, G, C, C, G, A, C, U. And this is a negative strand. So what is the point of doing it? Well, the genome of the positive strand is a positive strand, and you can't produce positive strands from a positive strand. You can't transcribe this and end up with that same sequence. The only way you can do it is by using the negative strand. So this is exactly what happens. This positive strand genome is transcribed as a negative strand. So here it is here. From that negative strand, we can produce a whole lot of copies of the positive strand. And those positive strands have two functions. The first is that they can function as messenger RNA, and they are translated to produce all of the viral proteins. But the second major function, of course, is we produce lots of copies of this to act as the genome. Once the viral proteins are complete and mature and they package themselves together into the capsids, then copies of the positive strand are packaged as the genome. So it has two functions. Okay, so now what about negative strand RNA viruses? Well, here's a negative strand genome. Now you can't produce copies of the negative strand from a negative strand. So in the same way, the negative strand is transcribed, and now it forms a positive strand. And that positive strand can be used to produce many, many copies of the genome there. And remember that this positive strand that we've transcribed from here, from the viral genes, the positive strand is used as messenger RNA. They'll produce lots of copies of it. And they use that to produce the viral proteins, but they also then transcribe it again in order to produce all the negative strand genome copies. These proteins package themselves together into the capsids. They get then the viral genome is then packaged into the capsid, and it's complete and can proceed. Okay, so I hope that makes that a little bit clearer. So I'm briefly going to run over these different strategies. We're going to first of all discuss DNA viruses because it's somewhat easier concept. Here's a really simple DNA virus, probably an adenovirus or something, I forget. No, it's HPV, papilloma virus. What does the virus need to do? First of all, it needs to attach. Then it needs to get into the cytoplasm. Once it's in the cytoplasm, the capsid here needs to be broken down, as you see here, and the viral DNA is released. That viral DNA makes its way to the nucleus and becomes integrated into the host genome. In the host genome, the genes read off two things. First of all, they read off lots of messenger RNA, which goes to the cytoplasm, which makes all of the viral proteins, the capsid proteins, etc., whatever else, other proteins it needs. The second thing is that this viral genome is replicated. It is replicated over and over and over and over again, so that it produces the copies of the genome. Once the capsids are mature, they assemble, and the genomes are packaged one to each capsid to produce the new viral particles. They only show one here, but in actual fact, there's many. For example, 300, 500, round about there, probably per cell, sometimes much more. Once that has happened, the virus leaves the cell by one means or another, either by being budded out or else by lysis of the cell. So that is replication of the DNA virus in animals. Think back, the replication in bacteria by bacteriophages was a little bit different. So now let's have a look. We're going to look first at RNA viruses, which are positive strands. We've already heard that, again, they have to attach, they have to get into the cell, the capsid is broken down, and the viral genome is released. And in the case of positive strands, that positive strand is replicated. It is not replicated, it is transcribed to a negative strand. And that negative strand is the functions so that many copies of the positive strand can be produced, which function as messenger RNA, but also end up getting packaged as the genome. The messenger RNA, when it's used as messenger RNA, it produces all the viral proteins. The capsid assembles, and the positive strand copies are packaged as the genome. Not difficult. So, the same thing in the negative strand RNA viruses. It's the very similar process: attachment, here entry into the cytoplasm. The capsids are disassembled. Now we have a negative strand, and what it does is produces positive strand in order to act as messenger RNA to produce the viral proteins. But in addition, from the positive strand, many copies of the negative strand are produced to act as the genome. The proteins are matured and assembled, and the negative strand genomes are packaged, and then the viral particle exits the cell by one means or another. Depends on the virus. So those are the DNA viruses. positive strand RNA viruses, negative strand RNA viruses, but there's one more that we need to talk about, and those are the retroviruses. Now, retroviruses are RNA viruses, but they have a positive strand which needs to be transcribed. I'll use the word transcribed. Needs to be transcribed to DNA, because these viruses function by integrating with the host genome. So they need to turn their RNA code into a DNA code, which is the reverse of our central dogma. Central dogma says DNA is transcribed to RNA. And this is the reverse. The RNA is going to be transcribed to DNA, and so we refer to that as reverse transcription. And here's the best known example, of course, of the retroviruses is HIV, human immunodeficiency virus, but there are a couple of others as well. The similar process takes place. Same thing as we've always seen. We have attachment, then entry into the cytoplasm. The whole structure of the virus breaks down. It's an envelope virus. So the envelope is lost, and the capsid is broken down, and the RNA, the positive strand viral RNA, is liberated. These viruses have their own enzyme that they carry with them in the virion, in the capsid, and they reverse transcribe their RNA, their positive strand RNA, to a double-stranded DNA strand. And that viral DNA then integrates itself into the host genome. In the case of HIV, there are a couple of cell types that it can infect, but the one which best characterized, you probably heard of, is the T helper cell, which is an essential part of the immune system. And in T helper cells, that section of viral DNA, which we call a provirus, remains silent. It is not transcribed until the cell itself is activated by the immune system. When it's activated in an immune response, then the genes are transcribed, and what they produce is, first of all, it's transcribed to messenger RNA, which is the same as positive strand. That messenger RNA then produces a very long strand of a protein, of polypeptides, when it is translated. But many copies are made so that they can be packaged as the genome, as the new positive strand genome. And that's exactly what happens. Eventually, the viral proteins assemble. They assemble into the capsid. They also reproduce the enzymes, which are proteins, which are also packaged together with the genome. And the virus then, the HIV virus then buds out of the cell membrane and acquires its envelope like that. Okay, so this just adds another step. It's almost like a DNA virus, excepting that the viral genome is actually a positive strand RNA. So it needs to be reverse transcribed to DNA, integrate, and then produce the new viral particles. Okay, so I hope that clarifies things a bit for you. So this shows you, this is actually HIV budding out of the cell. The one thing that they have not shown here is that the cell membrane here is modified by viral proteins. Viral proteins become embedded in the cell membrane, and those act as the viral spikes for recognition and attachment of the new viral particle to its host cells. This just shows HIV budding out and now invested with a membrane around it. Okay, so We're going to start moving on to considering viruses and disease. And the very first thing we need to consider are viral involvement in cancers. And this has been shown for several different types of cancer, that they are in fact caused by viruses. And they very often develop, the cancers develop long after an initial viral infection. It's important to note these are not contagious. Once the cancers themselves are not contagious. The cancers arise because of alterations to the host genome, because of the viral infection. So there are numerous types of cancers, but two groups that are particularly implicated in viral infection, by viral infection, are sarcomas, which are cancers of connective tissues, and adenocarcinomas, which are cancers of epithelial tissue, largely of the glands. So the way in which this, I'm only going to discuss this very, very simply. It's simply an introduction so that you know that this happens. We're not going to be discussing details of mechanisms. Within our genome, there are certain genes which are commonly implicated in cancerous tissue, in formation of cancerous tissue. And these are referred to as the oncogenes. And they are, remembering that cancer arises because of a dysregulation of growth and cell division. So many of the genes which are involved with growth and with cell division have the potential to be oncogenes. And they're called the oncogenes. Onco refers to the potential to create cancer. And they become oncogenes when their function is distorted. So, for example, if we were to fiddle around with any of the growth regulation genes, we might lose control over them. And if we fiddle with any of the cell division genes, the control over cell division, which is very carefully regulated by the cell and related to growth, then the cell might become cancerous and begin dividing uncontrollably, which is exactly what happens in the cancer. So what happens with the oncogenic viruses, the viruses which become involved with oncogenes, is that they get integrated into the host cell's DNA, into the genome, and they alter the function of the oncogenes. A simple example is papillomavirus, which, if you look at a wart, if you think about what a wart is, it's dysregulated growth of skin cells, but it is benign. It's, first of all, is benign. It doesn't spread. The wart itself doesn't spread, although the infection may spread. But it's also self-limiting. So it's a dysregulation, but it is completely benign. It's of no real import, and it's usually temporary. However, these same viruses, the papillomaviruses, can insert into the genome of cells like the cervix, the penis, and they can very much later induce changes in the regulation of those genes. So the genes actually become rogue genes, and the cell loses control of its division and growth functions. And that is almost by definition what a cancer is. So I'm sorry, I've done that twice. Okay, so just remember those terms, the oncogene, and remember that the oncogenes are those genes which have the potential, they're normal genes, they have the potential to cause cancer if their function is distorted by virus. So let's also think, in talking about that, about how many virus infections can in fact last for a very, very long time, often long after an acute episode. So we have already heard about herpes virus, and the way of herpes virus can actually become latent. It goes largely into the nerve cells and remains completely hidden, completely silent, until some environmental trigger causes another outbreak. And that is typical latency. That is, the host cells remain completely asymptomatic for very long periods of time, but then the virus can be reactivated. In other words, the viral genes are still present in the host. Viral particles may not be, and viral damage may not be, but the viral genes are there and can at some point start to be transcribed to produce new viral particles. And that is typical latency. But there is another situation with viruses, and that is where a viral infection actually persists over a very long time. And it does, very often what we see is that they will be doing a very little bit of damage to the cells. So for a very long time, there is no apparent damage. Damage is occurring, but it's occurring at a very slow rate and at an accumulating rate. The virus in the persistent infection remains active. It is actively replicated. In latency, it is not. In latency, it remains completely silent. And there is one particular disease which is really famous for doing this, and that is measles. And measles in children causes an acute infection, which we'll discuss a little bit more a little bit later. But it causes an acute illness. But in some cases, fortunately, this is extremely rare. But in some cases, the infection is never completely wiped out by the immune system. And instead, it persists in neural tissue over a very long period of time, of about a decade or so, and then manifests itself as this terrible disease, subacute sclerosing panencephalitis. And it is quite literally measles of the brain. And it results in loss of neural tissue, and ultimately it is fatal. And once it commences, it is not treatable. So this is another very good reason for getting measles vaccine, because this, although it's very, very rare, it is a terrifying disease. So there are a number of viruses that do this sort of thing. They're either latent. These three here, the herpes viruses you already know about. Second of all, this one here, the HTLV1 and 2, these are retroviruses. And these can do the same thing. There is an initial infection and then a long period of latency. And only then, later in life, do they reactivate to produce leukemia. The other one you have heard about is shingles. This is varicella virus, and that is the virus that causes chickenpox. And of course, chickenpox, there's an initial acute illness with skin lesions, usually in children, can occur in adults if they haven't had it before. And if you have it as a child, the virus behaves very like a herpes virus and goes into the nerve cells to become latent. In many people, it never reactivates. There's no inevitability of it reactivating, but it can reactivate, and it then does so as shingles, a very, very painful rash, either on the face or the trunk usually, although it can be elsewhere. So persistent viruses, those that cause a long-term, low-grade illness. The papillomavirus causes its cancers by continual infestation, especially of the cervix. Again, there's no inevitability about it. Just because you've had genital warts, for example, does not mean to say you are definitely going to get cervical cancer. And likewise, people who have cervical cancer may be completely unaware of the fact that they were once infected with papillomavirus. HIV, AIDS, that is also a persistent infection because they infect the cells of the immune system and of the nervous system, but the immune system is more impactful. And over a very long time, they degrade the T cells, especially in the immune system. The only other one that I want to mention here is our subacute sclerosing panencephalitis. And that is an important one to remember. That's by measles virus. Okay, so just a couple of other sort of weird things. They're literally just things. They're not living organisms or anything. And I'll just mention a couple of wandering biomolecules that can cause illness. And the most important one are these, the prions. And prions are a protein. What they actually are a distortion of natural proteins, particularly of the nervous system. So they have a counterpart which is normal, which occurs in the nervous system. And these are, you can't really say that they're mutated. They have changed form. And when they get into a cell, they start to induce all the normal protein that resemble them to become like them. And they in turn become infectious. And these are called prions, and that stands for proteinaceous infectious particles. And they are extremely dangerous. They are inherited, and they're also transmissible. And you can get them by eating food with prions in them. And then you can also be infected with them if you, for instance, get a transplant or surgical instruments have been used on somebody with the prions and then used on you. And one of the terrible things about them is they're almost impossible to disinfect against. And so they cause a group of diseases called the spongiform encephalopathies. And the one that you've probably heard of is mad cow disease. Mad cow, because the cows that are infected with it begin to behave in a very strange way. They begin, their locomotion is affected and their behavior is affected. So mad cow disease was really important because one of the things about the prions is that they can be acquired through ingestion. And in Britain and in Europe, there was an outbreak of mad cow disease. It's actually called, in human beings, called Creutzfeldt-Jakob disease. And Creutzfeldt-Jakob disease was acquired by numerous people through eating hamburger. And what had happened was farmers, because they saw their cattle becoming ill, if they saw their cattle becoming ill, they would sacrifice them and send them to the meat market. And not knowing, of course, that they were transmitting infectious disease. But the hamburger, the cows entered the meat train and they ended up as hamburger. People ate it. The prions are not degraded by heat, and people acquired it through ingestion. And then once that was discovered, that was the origin, very strict new regulations were put in for inspecting cattle while they were still alive to ensure that they do not contribute prions to the meat market. Now, I would want to tell you one other interesting thing. Creutzfeldt-Jakob disease was actually described in cannibals and in Papua New Guinea. And that was where it was first noticed. It was a common disease amongst people who ate one another. or ate their enemies. In the 19th century, they was identified, and of course, because what was happening was that people were infected and were then killed and eaten. It was a rich source of prions to the people who ate them. There are a number of other diseases. This is the only one I would want you to remember, and that is scrapie and mad cow disease. Both contribute to the occurrences of Creutzfeldt-Jakob disease, because of course they are both eaten by human beings. So those are the prions. All they are is a protein. That's it. They're not alive. They have nothing other than the fact that they are a distorted form of a naturally occurring protein, and then induce those natural proteins to alter their form to the prion form. Just a couple of other things to mention. The first is we've talked only about bacterial and animal viruses, but I do want to tell you that plant viruses are enormously important, and they contribute to massive agricultural loss every year. Now, one of the things about plant viruses is that plants are very well protected against viruses by their cell wall, because they have a thick cellulose cell wall, and it's very difficult for viruses to enter that. If you just drop a virus onto a leaf, nothing will happen because the virus can't attach to a living cell. The cells are protected. But if that cell wall is breached, if the plant is wounded in any way, then viruses can get in. And for that reason, viruses are most commonly transmitted amongst plants by biting insects, by insects which feed on the plant, breach the cell wall into the cells, and of course can transmit the virus that way. The insects now are acting as a plant virus vector, and they will transmit the virus like that. The other really strange thing, somewhat like the prions, are the viroids. And viroids are, fortunately, they only occur in plants, and they are short little pieces of RNA, just a short bit of RNA, enough genes to alter the function of plant cells so that they produce masses of this RNA. They're very, very strange. Fortunately, they only cause plant disease, not human. So the way in which the viruses cause disease, apart from the oncogenic viruses, the viruses, once they infect a host cell, they're going to radically affect the cell function, because what they're doing is they're taking over all of the metabolism and machinery of the cell, all its transcription, translation, many metabolic functions, and they ultimately cause cell to start to dysfunction. And they usually end up causing cell death by the time viral particles begin to be released. So they drastically alter the metabolic function of the organism that they're infecting in general. And in addition, the organism has to deal with a large amount of waste material from dead and dying cells, and that sort of thing. And the other thing is that they very frequently produce overreaction of the immune system, and they cause the immune system to actually lose control of the immune response. And you may have heard of this in the COVID pandemic. Many people died as a result of what were called cytokine storms. What a cytokine is an immune system hormone. It is the way in which immune cells talk to one another and influence the activity of different branches of the immune system. And once that communication begins to break down, very, very serious consequences arise. In particular, that the symptoms of inflammation that we talked about, for instance, when we talked about sepsis and septic shock. Similar things start to happen with viral infections. The other thing is that viruses in damaging tissues make them very vulnerable. They make them vulnerable to secondary infections, particularly by bacteria. And so, for example, respiratory tract infections, especially lower respiratory tract infections, predispose to pneumonias and things like that. And in fact, in respiratory infections, the virus itself may cause a pneumonia, an inflammation of lung tissue, which is referred to as a viral pneumonia. Okay, so let's start to have a look at some particular diseases. We're going to discuss diseases of the upper respiratory tract, first of all. And these, of course, are viral infections, which are very, very familiar to all of us. And the one that we instinctively think about is the common cold. And common cold is very, very common. In fact, most people record that they have about 2 colds a year. And there is a famous passage in the Quran in which Muhammad, the prophet, says that people of Medina said that I was mad, even though I had 2 colds that year. Now, that reflects a very old belief that having colds was in fact healthy. And that if you didn't have colds, you were predisposed to nervous dispositions. And there is some truth to that, because it has been shown that having a cold is a very good stress reliever. One of the reasons being, I think, that it's simply a good excuse to go to bed, go and lie down for a day or two, and don't worry about anything else. Why? I'm sick. And I really think that there is some truth to that. So the symptoms of the common cold is an upper respiratory tract infection. That's really important to remember. So you may have a sore throat, runny nose. That's the primary symptom. If there is a fever, the fever is very mild. If you have a headache, the headache is usually very mild, and the headache may in fact be because of inflammation of the sinuses more than anything else. You might have muscle ache, but again, it's not usually very severe. And another symptom, is very important symptom, is that you feel fatigued. And again, that is to make you go to bed, make you go and lie down. It is actually part of your immune response. And in fact, many of these symptoms that we see here, the fever, the muscle ache, the fatigue, these are all the result of immune responses to the presence of the virus, not to the virus itself. Although we always associate those 2 words, coughs and colds, in actual fact, the cough in common cold is quite rare. And it will usually arise because of a secondary bacterial infection, which spreads to the bronchi. So real important, I'm emphasizing this mild headache, mild fever, mild muscle aches, that sort of thing, because when we start to talk about influenza, we're going to see a different picture. The other characteristic of the common cold is there is a long prodromal phase. You have days when you kind of feel that you're going to get a cold, but before it actually arrives, before you develop your signs and symptoms in full-blown form. So most of the upper respiratory tract infections are viral. They are very seldom bacterial. We will hear about some when we talk about bacterial disease. But here is a very common cause of the common cold, and that is rhinovirus. And rhino refers to the nose. That's why rhino is called a rhino. So rhinovirus infections, they occur in the upper respiratory tract, because the upper respiratory tract is actually slightly cooler than the rest of the body. It stands to reason. We've got a very big evaporative surface in our noses, and the temperature of the nasal tissue tends to be lower, about 33 degrees Celsius. And that is one of the reasons why the onset of a fever is actually beneficial in a cold, because it raises the temperature, and these viruses don't function well at a higher temperature. The common cold is extremely easily spread. It is spread largely by airborne respiratory droplets, but it is also spread by fomites. So the subway pole, table, lunch table, these viruses, many of them can last for a very long time on a surface like that. But the one thing to remember during cold season is to learn not to touch your face, because our hands pick up the virus. And if we then touch our face, especially anywhere near the eyes are a very common source of infection. The cold virus is commonly transmitted to the eyes first, then to the upper respiratory tract through the tear ducts. Okay, here's another cold virus. This is adenovirus, and I mentioned this before. You can see here, it's that polyhedral capsid with these distinct, this one in particular has these distinct spikes. Not all adenoviruses do. And this is another common cause of a cold. But the adenoviruses can be a little bit more severe than some of the others, because some of them do produce quite high fever. And they're also associated with a greater number of throat infections. So that's called sore throat. The proper name is pharyngitis. So they're common causes of pharyngitis and cough. And they can sometimes lead to a viral pneumonia. Most of the other cold viruses do not do any of those things. The adenoviruses are known for it. So the upper respiratory tract infections are usually relatively mild, self-limiting, and seldom need medical attention. The one thing I would mention, though, is our instinct is to rush off and start swallowing Dayquil and NyQuil and aspirin and all that sort of thing. Is probably not a good idea, because what you're doing is you're interfering with the immune response. Many of those symptoms that we find unpleasant are because of the immune system taking care of the infection. So certainly if symptoms go out of control, if the temperature goes very high, or people start to get confused or anything like that, then medical attention needs to be sought, even for upper respiratory tract infection. However, here's a disease which really needs careful watching if and whenever you have it, and that is influenza. And influenza can be an extremely severe disease, and the reason for that is that it's actually a disease which tends to move to the lower respiratory tract, to the bronchi, and then to the lungs. Influenza is also a very, very highly communicable disease, and it is a really potentially very serious illness. When people do have influenza, it should not at all be confused with the common cold. This is a different set of viruses, and the signs and symptoms are, in fact, different if you know what to look for. Colds and flu get confused because both of them have upper respiratory tract signs and symptoms. So both can produce rhinitis, runny nose. Both can produce sore throat, even without a bacterial infection. So they tend to be confused, but there are some distinct differences in the symptomology. And the first thing is that in influenza, the onset is extremely sudden. There is a very, very short prodromal phase, if at all. And very often the first sign, the first symptom that a person will feel if they're getting influenza is a blinding headache. And a severe headache is one of the hallmarks of influenza infection. And the second thing is that influenza tends to produce a much higher temperature and more severe body aches, joint aches, muscle aches as well. Thirdly, it tends to move to the lower respiratory tract, and it produces cough, copious production of mucus, etc., congestion, all the rest of it. But depending on the flu strain, the flu virus has the potential to be extremely serious, if not fatal. And influenza epidemics always carry with them increased mortality, especially amongst the elderly and also amongst the very young, amongst young children. That is the normal sort of distribution of mortality. But influenza, in addition, in certain strains and in certain circumstances, has its highest mortality amongst young people. And that is because of its propensity for causing a massive immune response. So people with the most healthy immune response tend to react the worst to certain flu strains. And that was the case in the 1918 flu epidemic and in several other flu epidemics that have moved around the world. The mortality often is associated with overreaction of the immune system. So influenza is very communicable, but it's mostly by airborne respiratory droplets, less by a fomite. And one of the problems with influenza virus is it changes the whole time. And it changes in two forms, as we'll hear in a minute. The one type of change is a slow change, an accumulation of mutations over a period of a year or so, a year or two, so, which makes the virus invisible to the immune system of people who've had influenza even the year before. Their immune systems don't recognize the new flu. That's the one problem. The second problem is that every so often, more on a decadal timescale, entirely new flu viruses arise. And I'll explain exactly what the mechanism for that is. And when that happens, the new flu, it's almost like a new flu species, will erupt into the population, not only human population. It will erupt into the susceptible populations and cause massive epidemics and massive amount of morbidity. The influenza viruses are in fact zoonotic diseases. And that is because there are three major reservoirs for the virus, and they are human beings, pigs, and poultry. And the virus can move between those three reservoirs. So it is our most important zoonotic disease and one of the most important human diseases because it has a huge influence on human mortality. It's been recorded since about 1510 in a recognizable description of the symptoms. It may have occurred before 1510, but not being recognized as an illness. But it was first, we can see the symptoms, signs and symptoms described as early as 1510. So it shares those upper respiratory tract signs and symptoms with the common cold. But again, it has much higher incidence of fever, sudden onset of headache, often quite severe fatigue, muscle, and joint pain. And then the most dangerously, or in terms of its immediate symptomology, lower respiratory tract symptoms of cough, inflammation of the lungs, and then viral pneumonia, and then predisposition to severe bacterial pneumonias as well. And it's sudden onset, which is so marked, and it also implies that people who suddenly go down with flu may have had a long preceding period of being infectious, which they didn't know. Okay, so it's an enveloped virus, and on the surface of the envelope, there are two kinds of spike. And the first are called the hemagglutinin spikes, or the H spikes there. Now, these are complex proteins, and the hemagglutinins are attachment proteins and also assist in penetration into the cytoplasm of the host cell. And we recognize 17 kinds of hemagglutinin spike, H1 to H17. The other kind of spike is called a neuraminidase spike, or N spikes. And these are more concerned with the emergence of the virus from the host cell, not with its entry. And there are 10 kinds of N. More will surely be discovered as time passes. And these the N spikes are numbered N1 to N10. And any influenza virus can be characterized by its H and N proteins. So that's why you will hear a flu virus referred to as being an H1, for example, an H1N1 virus, or any other combination of these. It has a very strange capsid structure. And first of all, there is an outer protein shell here, which is called the matrix protein. Here on the outside, that's the envelope there with the spikes. Here, this is called the matrix protein. It's not the capsids, because in fact, they have, they have 8 capsids. 1, 2, 3, 4, 5, 6, 7, 8. They have 8 capsids, and their genome is split between those 8 capsids. It's like 8 little miniature viruses contained in a single virus, each of them with a piece of the genome in them, like that. And each of those are carrying the viral genes, which are then going to be replicated and everything by the host cell. So each of these carries characteristics of the virus, and there are in fact 3 kinds of flu that are recognized, but this one, influenza A, is the one that is most important. This is the one which causes most epidemics, and this is the one which comes every year, which we see every year. This one, influenza B, it does come to, it's very uncommon though, and it's less severe than any of the A's. Influenza C, it's really mild and does not cause, it's always localized. It doesn't cause epidemics. Now, they are associated with influenza in addition to all the things I've told you about. There are some other syndromes which are associated. The first is Guillain-Barré syndrome. And the Guillain-Barré syndrome affects the nerves and causes a paralytic illness, sometimes self-limiting, but sometimes a long duration. It's rare. It's a rare complication of influenza. The one that all of you who have children or will have children should be aware of is Reye's syndrome. And Reye's syndrome occurs in children who have been given aspirin, usually as a result of having a cold or influenza. And this can be an extremely severe illness. So you need to be very careful. Don't give children aspirin. Acetaminophen, stuff like that, they can have, but aspirin is counterindicated in young children. Okay, so I'm just very briefly going to explain to you that those 8 little capsules of the genome are the source of much problem. And the reason for that is that there are 2 ways in which the influenza virus can change. The first is that there is just, as the virus moves through epidemic, as it moves from person to person, and ultimately from country to country, the influenza virus can accumulate small changes to the genomic structure. So small mutations, and these are usually result in changes to the H spikes, and may also affect the N spikes, of course, but it's the H spikes which are particularly important because they govern attachment and entry. And these small changes, but they are sufficient that these spikes are the things that our immune system recognizes. So any changes to the spikes will affect the immune system's ability to recognize the virus. And if they accumulate sufficient changes, the virus effectively becomes invisible. And this happens over a yearly, 2-yearly basis. And what happens is it usually begins in one part of the year, and it takes some time for it to make its way around the globe. And these are the cause of the yearly outbreaks of influenza. So what the authorities do is they go around the world and they sample influenza outbreaks. They sample the virus, and they try to identify the top 3 strains of the virus every year, which might cause epidemic elsewhere. And those are the 3 viruses they choose to do make vaccines for. That's when you get your yearly flu vaccine, that is what is happening. You're being given the most likely candidates for epidemic influenza. The flu vaccine, by the way, is not a living vaccine. It's not a modified virus or anything like that. It's just particles of the viral protein. That's all. It's not. It cannot cause flu in you if you have the virus, if you have the vaccine. So this slow change is called antigenic drift. Antigenic refers to the ability of the immune system to recognize. The immune system recognizes these, which are antigens on the outside. So this is antigenic drift, and it's the cause of our yearly not so serious epidemics. We always have some immunity. We always carry over a bit of immunity, so from year to year, but it's not sufficient to prevent us from being infected by these new antigenic strains. However, here's the big problem. Remembering that the genome is cut up into 8 pieces. There is a possibility for influenza to infect a poultry, pigs, and humans. And when they do so, there is a possibility, let's say, that there are two different flu strains. One which is infecting birds, and then there's another virus which is infecting humans. Remembering that we have a third reservoir, it is possible, for example, for a pig to acquire both the avian influenza and the human influenza, and to reassort the genomes of those in a new virus. And this is called antigenic shift. And it is extremely dangerous, because what has happened is you've effectively created an entirely new influenza virus. You've taken bits of this poultry, you've taken this human, you've reassorted them in pig, and you've produced a new virus. And that can jump to humans. In addition, any one of these viruses, human or the avian, can move through pigs and be adapted in pigs, can be changed in pigs, and then be passed to humans. In this way, for example, we acquired H1N1 influenza virus, and that came directly from poultry via pigs to humans. And third, we can have a jump directly from birds to humans as well, and acquire bird flu virus, which behaves completely differently. These are all very dangerous situations, because it means that entering into the human population is a virus, however it's been arrived at, which is completely different to anything else we've seen through antigenic drift. And this is the cause of major epidemics of influenza. And the greatest of all of those epidemics was 1918. And that epidemic resulted in millions and millions of deaths, far more deaths than COVID-19, and was a major event on a global scale that rivaled the COVID-19 epidemic. Okay, so I'm going to end off there. I will, as usual, I will repeat a little bit in class for you of how we ended up. But just remember, please, that you have a test next Monday. There's a lot of material up on Canvas for you, including now questions regarding epidemiology, and other study guides have been put up as well. Okay, so good luck with your studying, and I'll see most of you next Monday. See some people tomorrow, but I'll see most of you next Monday. Okay.